period, dry fallow period. Very often you see this as dry, blackish, brown patches on the soil as well as adhering to various weeds and rice plants. Now, what is the outline case of the fertilizers this sequence can change? Now, having seen what is happening in nature, then we get on to what we as humans try to intervene and manipulate the system to our advantage. And such method, Raman was one of the pioneers in that, and he called this process alkalization. Now, in Sri Lanka also, we conducted a survey of nitrogen fixing blueprints present in rice soils. Now here we got this situation where from different localities of rice nostoc, Anabina, Calopix, Cygonium, Fischerella, and here we have the different localities. I don't think these names would mean anything to you unless there's somebody who has visited Sri Lanka regularly. We have the highest frequency from that culture as nostoc, and the nostoc, most of the nostoc propagules seem to grow up. But when you observe in nature, we don't see so much on nostoc right in the rice field. But the propagules are there, given the right conditions they seem to come up. But in terms of frequency, they appear to be the most common. And Roger and Grant also resist or withstand algal grazing by the microfauna. And that also may reflect a high presence of nostoc in soil that you have. And this is the normal insecticide that is used in rice to control all mildew and such rice stones. There's nothing new to the rice cultivation. A last question?
How extensive is Azolla used in your country compared to other cyanobacteria? And the second is, do you have a problem of biomass building up in the field and causing some problems?
The rice
No, when you use Azolla instead of just the other kind of cyanobacteria. Do you have biomass of Azolla building up into the field?
Okay. Yeah. First question is whether to what extent farmers are using Azolla. Well, there's no promotion at all, either by the government or by any other sector. The farmers on their own have now caught up in the wet zone, Lokani wet zone of Sri Lanka, and they are going in about 500 to 750 hectares depending on the season. That's the extension situation now, without any encouragement. The other question is how much of biomass is left. Now,
Foundation and Professor Menon recorded that a huge amount of nitrogen is fixed biologically as compared to the chemical nitrogen fertilizers produced in factories. We had a good presentation from Dr. Surya, and he highlighted that it is difficult to build the introduce algal population in the rice field. But there is nothing to panic about this. After all, it's a biological system and which is affected by a variety of factors. We have initiated, we may claim ourselves as a pioneer. My teacher and predecessor, Dr. G.S. Venkatan, who was honored yesterday, had initiated in 1960 this work. We also face the problems, I think you can start slides also. We also face the problems, and we have tried to understand the region. Actually, we were advocating the saving of in terms of nitrogen so far. But the last 5 years we have changed the strategy, 5 to 7 years we have changed the strategy. It's not only the nitrogen, what other else it contributes to the productivity of the field or to the increase in the crop yield, we have tried to analyze. So my talk will be divided in two parts: the earlier technology which was developed, and then how to improve the existing technology that it can go to the commercial scale. We have been partially successful in this attempt, and I will run across two slides. Of course, Dr. Surya had shown this is only to depict how much fertilizer or how much biological nitrogen fixation takes place. This is a bit old, shows 175 million tons, but now it is around 200. What?
Do you have some slides also? First, you said grazing pressure.
Grazing pressure on cyanobacteria.
Yes. And second part of the question?
Is there active microbial food web. That means
Yes.
Anabena
Yeah.
and protozoans, rotifers or something else.
That's true. Actually, we have observed in the field as well as in the production units that the population of grazers which feed on the alga or algae is there, and this problem is more where water is always standing. Where water is alternately, not standing, this is just a moist surface and submerged alternate, there the grazing population was very low. Though we have not exactly estimated how much biomass is consumed by them. As in the earlier talk it was referred, that we do control these grazers, but we control only the production units. That was the problem earlier existing when it was produced in the open area. But that grazing problem is not coming now, at least in the production unit, when we are doing in the polyhouses. But this problem do exist under field condition, and it is difficult to check, because if we are going to control that population, then probably we are adding another factor which will add to the cost of production. And this is not a universal problem in all the places. As I told you, this is the problem existing where always water remains standing. So probably that aspect we will look into detail. We have not worked out on that.
I mean, it is not only a problem to keep the cyanobacteria in the field, but I think it is a very important thing also for remineralization. If this microbial food web is working very well in such a field, you have a very enhanced remineralization of the cyanobacteria. And if it is not very working, it will be stopped or something changed. And so I think you should look on this microbial food web in the field, and you should try to find out what is the cycle running there.
Yeah. Another question? Yes.
Yes, about the soil pH, you mentioned the 7.3, slightly alkaline, but where in Nepal we have slightly acidic soil, 4.5 to say 6.7. So what could be could you suggest something suitable for pH slightly say acidic, or sometimes it is more acidic, 4.5. So what to do?
Yeah, you are very right, Dr. Tushar. The acidity do occur also in India, particularly in Kerala and northeastern states, and as well as it was referred from Ceylon also. We have some strains which are acid.
Well, that point was highlighted by Professor Kolasuri also, saying non-stop and
Glutetia for the
see, to avoid grazers, we cannot take a form which do not have faster growth rate, which do not have high nitrogen fixation ability. So we have to take both the points into consideration: nitrogen fixation rate, growth rate, as well as this. They are protected from the grazer by their unlikely they are fed to the grazer. We have some strains actually in this work I have not highlighted. They are not preferred as a food by these insects, but then they are not good nitrogen fixer. That's why we have not introduced in the program. Probably in future we may come some forms which are not liked by this grazer. That will add, I think, that will be better.
Major problem is establishing.
Let's continue that discussion later. I think at lunch or during lunch. I'd like to go on to the next talk by Dr. Ann Rye, Cyanobacterial Nitrogen Metabolism, Relevance in Agricultural Biotechnology.
The cyanobacterial nitrogen metabolism relevance in agriculture biotechnology. Now this I put only because this is a biotechnology conference. The work we are doing is basically on cyanobacterial nitrogen metabolism, mostly on symbiotic systems, and also on parallel work we have done on free living. This is basically because this has given us a lot of information to apply from symbiotic systems to free living and vice versa, to sort out some of the mysteries as far as we are concerned in terms of nitrogen metabolism in cyanobacteria. Now, the Before we go to the main topic, I would like to give, I'm restricting my talk to mostly nitrate metabolism and ammonia metabolism. Before I go into the talk, I just want to remember that nitrogen fixation vis-a-vis nitrate metabolism, both of these processes will generate ammonia into the cell. What is the cost like? You need not read all of it. You can see that specific reaction rates, the nitrate systems are much faster. But the energy consumption, if you see, 960 kilojoule per mole ammonia produced, that is in the cell, and compared to 700, which is from nitrate to ammonia, including the transport and reduction of it. The turnover rates also you can keep in mind that the nitrogenase from all these points of view is rather expensive. So this is why the N2 is the last resort for the organism as far as its choice is concerned. If it gets ammonia, that is the first choice. If it gets nitrate, it will be second choice, and the last one is N2 when nothing is available. That's why you see repression by ammonium voltage are repressed by ammonium as well as inhibited. Coming back to the talk, the free living cyanobacteria, I have seen several slides this morning and also yesterday. You are familiar with heterocystus ones, non-heterocystus ones, free living ones. I would just like to show you some of the symbiotic ones, not the cyanobacteria, but what they look like in symbiosis. So if I can run through first few slides.
Talk is open for discussion.
Applying this ammonium fertilizer, say urea in the field, I have studied in situ, and in both cases the nitrogenase was reduced, no doubt, but it was not like that the completely inhibition was there. And within 4 days in the soil system, cyanobacteria regained the process. Whether it is calcium nitrate source, whether it is the urea source, inhibition was almost similar in the soil system. What may be the reason you think? Because in the pure culture you are observing like this, but in the soil system it's quite different.
The answer is that I don't know because I never worked in the field. So
the answer is that whatever form you give,
so that means it's in the field, it's nitrogen is always
The
taken.
rice crop, the ammoniacal form of nitrogenous fertilizers are applied. There are two basic reasons. Rice take in the form of ammonia. Secondly, any nitrate fertilizer nitrogen, there is a loss of leaching. Secondly, the nitrate also has to be converted into ammonia. So the additional requirement of energy and the metabolic process do
I
take
think it's
place.
cheaper than that.
So the ammoniacal form of nitrogen becomes easy to apply, and that's practice. The farmers are not using nitrate form of nitrogen in rice cultivation particularly. And what the Dr. Jha referred, I referred in my talk also that soil is a very heavy buffering system. 4 days, as you said, the nitrogenase activity is regained by the organism. That's what I said. Immediately when the nitrogen fertilizers are applied, if they are in direct contact, the nitrogen fixation is inhibited. But as you yourself have observed that after 4th day or 5th day, the nitrogenase activity is regained. That's because the dilution effect of the applied nitrogen.
The question is whether it is a calcium nitrate.
We'll discuss it.
Okay, so we'll continue that discussion later.
And then come to you.
I think I'd like to make one
I
other
never
point.
seen the field and the other
We will work together. No problem.
down there.
I'd like to make one other point, because there's something we have published that now is different for different organisms. And that is the mention in the talk that nitrogenase in Anabina is restricted to heterocysts. That's true in Anabina 7120. But in Anabina variabilis, ATCC 29413, it's now been shown very, very clearly that under anaerobic conditions, there's a completely different molybdenum-based nitrogenase, which is active in all of the cells. Whereas the original molybdenum-containing nitrogenase is again specific only to the heterocyst. So we go on now to the last talk of the morning. Dr. Tishner will speak on nitrogen metabolism of non-nitrogen-fixing cyanobacteria and green algae special features.
The University of Göttingen by presenting the picture of the most famous member of the university, which is a goose girl. This goose girl is standing on a fountain in front of the old town hall, and all PhD students, after their final exam, they have to climb this fountain, and the male students will kiss her, and the female will place some flowers in a basket. So that's just the important member of the Göttingen University. And so you can see that the light intensity is important, and we've got a pretty early saturation to the 30% of 0.1 or 0.4 times 10 to the 3 watt per square meter. In the next slide, you can see that the uptake rate develops if the cells are cultivated in a 12 to 12 light dark chain. I want to mention these cells are not really synchronized. It's very difficult to synchronize these cells, as Dr. Kautik knew. He did this, he tried it at Göttingen. And but what we can see here is that after 2 hours of illumination, the highest uptake rate is obtained, and later on in the cell cycle, the uptake rate of nitrate is decreased. May I have the next slide, please? Depending on temperature, and this is the Arrhenius blood, shows that at higher temperatures than 41 degrees, you've got a kind of a membrane phase separation, and the uptake is not longer occurring. From this slope, you can calculate the Q10, which is in the range of 3.6, which indicates nitrate uptake to be an activated process, an ATP-dependent process. May I have the next, please?
Now, whether you use the same media used for culturing the higher plant photographs.
My scholar, Mr. Subramanian, to present a memento to Dr. Kautik. I would like to inform the delegates who have paid money for registration, journal issue, field trip and so on, to collect the receipts for the same from the volunteers sitting in the foyer. And those who are presenting papers in the poster session in the evening are requested to make use of the lunch interval to go and paste their posters, so that otherwise you may not find time to stick your posters. And just before the poster session, that's after the tea break, we will have a very short video program. The Department of Science and the Department of Biotechnology, Government of India, has prepared a 20-minute video program on biofertilizers. We want to exhibit that in this hall before we go on to the poster session, probably followed by a 10-minute video program which Dr. Gigi Boyd insists that we should see on Spiroline. So after these 30 minutes of eating up of the poster session time, I think we can compensate it. Instead of closing at 6:15, we can take the poster session up to 6:40. Then we have to board the vehicles available to go for our cultural program at another place in the city. So now we can adjourn for lunch.
It is on a continental
This is an opposite vegetarian. And this is continental. No hot food. No hot food. That's fine. That's fine.
We tried when
People are so strict to their material, like the people, you know. If someone is very So can we request a reprint when you publish this work? I don't know when I something like Good. When you are going to my property tomorrow, and I am looking
growth of spirulina. This is a Dunaliella plant in California, growing Dunaliella using paddle wheel ponds, chlorella production, and so on around the world. This is probably the most common algal production system for the commercial production of microalgae. The alternative system that we have, next slide please, is the extensive system which is being used in Australia for the culture of Dunaliella again, This is the system that I grew up with, if you like, and I'm still growing up with. To give you an idea of the scale of this culture system, each of these ponds here is 5 hectares in area. This pond is 50 hectares in area. This is actually a 2-year-old picture now. The plant now has another 2 of these ponds and a 100-hectare pond as well. So this is extensive, unmixed, shallow pond culture. There are 2 plants like this in Australia producing most of the beta-carotene from Dunaliella commercially. They've been in operation for almost 8 years now, and it's a very large industry. This works for this alga in Australia. It doesn't work anywhere else. So where do we go after these systems? We can grow spirulina, we can grow Dunaliella, we can grow chlorella, we can grow Cyanobacterial commercially, and that's about it. On a large scale, and to be a large scale is 1 million litres or something like that, or I usually think more in tons of algae. For most of the other algae, we have problems of contamination, controlling the growth, etc. So we tend to need a closed system. So the limits we have to the open cultivation systems, the ponds, the paddle wheels, and so on, is firstly, they're very inefficient as far as light goes. Usually you've got a water depth of 20 to 30 centimetres, or even deeper, so light penetration is limiting. You've got no temperature control, or very little temperature control. You've got a contamination problem. It's open to the air. The contamination may be other organisms, or it may be heavy metals, it may be pesticides, it may be, well, in one of our open ponds we had kangaroos in it, so this is a contamination problem. And you have a problem, again, from the efficiency of the culture, which relates to the turbulence in the system. They're very unevenly mixed. These paddle wheel ponds are very badly mixed systems. The sort of culture system you have in the laboratory, which is reasonably well mixed and so on, when you go larger, you don't have that problem. All of these things limit your productivity and limit the species that you can grow. The answer to that, of course, has been around for a long time, since the 1950s. We need a closed culture system. And since the 1950s, people have been trying to find a commercially viable closed culture system. Next slide. The sort of photobioreactor systems, the closed system, 50 to 150 centimeters. Then we have plate reactors. There's a whole range of types of deals for maximum growth. And then the doubling time.
How much time it takes to reach that density? That's the question. With optimum conditions which you are giving light, temperature.
Spirulina, I can't tell you because I didn't do the work.
Spirulina.
But for the isophysis, for instance, takes you about a week to get up to operational density, but then you work at continuous culture. For 2 reasons. The cells are very fragile, and secondly, the tubing absorbs UV light, and Dunaliella needs the UV light to get the high beta-carotene content. So it grows in there, yes, but not as a commercial system, forget it. No.
Another question, do you find this problem of forming in it?
And that really relates to if the algae are growing happily, then you don't have a foaming problem. If they're an alga like Porphyridium, which produces a lot of extracellular polysaccharide, you have a major thing.
How about the oxygen? Oxygen you said.
It's a problem because you build it up in the tubes and then you strip it out. It is the one area, and the tube length is a compromise to a longer tube is cheaper, but you have your productivity.
What is the tube length you have?
100 meters.
100 meters.
Is one cycle.
1 cycle.
100 meters.
As you have changed the population density, have you changed the concentration of the medium?
We just changed the density by harvesting in some of the cells and
Cleaning of this. You do not this one.
right. Yes.
Because it's a very vital.
Yes. It depends on the species. There are 2 ways to clean them if they do stick, or 3 ways. With the Cleotrichia, what you do is you take your shoe off and you hit the outside of the tubing with the shoe. I have yet to make an automatic process. You can also put small beads on the inside which knock the algae off. So you can add some small beads. We're still trying to make that work better, again to automate it on a larger scale. But for most of those species we don't need to, which is much better.
The writer of the United States says that getting a product out of algae is more expensive than getting a product
from any other organism. What is your opinion? Same product from any other organism. What is your opinion?
He's right. I fully agree. And the problem is because they need light. And to build a reactor where you get the light to the organism.
Ladies and gentlemen, it's nice to be back in India again for the 20th time. Sometime, at least 500 years ago, the Aztec Indians discovered that eating the Cyanobacterium Biotechnology, which they called equiplateral, and we called spirulina, gave them the strength they needed for their mode of life at 2,500 meters altitude. One legend is that fresh fish from the ocean was carried up the mountains to feed the Aztec kings at Tenochtitlan, 200. Just having witnessed the 1996 Olympic Games at Atlanta, it should be no surprise to learn that both Chinese and Cuban athletes ate spirulina during their training and in terms of gold per inhabitant. The farm depends upon the location, the amount of spirulina to be produced, the market for which it is intended, and the amount of money available to build the farm. Starting with family-sized units, as one needs about 1 square meter in batch or discontinuous culture, are supplied in prepared doses. And all the farmer has to do is harvest every day or so until the culture dies for lack of nutrients. Or the chemicals absorbed by the algae can be replaced at each harvest, and the process can be made continuous. Permit me, please, to use the word algae because the Cyanobacterium are the leader of bicarbonate to assure alkalinity, a source of fixed nitrogen, nitrate being the most stable, easy to use, and the most expensive, 8.5 and 11. Daytime water temperature from 30 to 38 degrees C, daytime stirring, and regular harvesting in replacement of the elements taken up by the algae. The next step, inspired by the spirulina E lesser to mingle, which leaves its ejections in the water to be recycled by bacteria. The system ferments village wastes in a biogas plant, producing methane for cooking. CO2 spirulina gets kwashiorkor children back on their feet and strengthens their immune systems, while the empty finders attached to wood frames or surrounded by earthen banks also have been used. Stirring is accomplished by paddle wheels driven by electric motors, energized by solar photovoltaic, as emphasized spirulina farms. Today, there are about 30 million children in the world who are at high risk of dying from malnutrition. It has been shown that given these children 10 grams of spirulina a day, plus a minimum of cereal calories, that can save their lives and give them normal health. In order to do this, we would need to produce 109,500 tons a year. Quite a jump from today's 2,000 tons. Obviously, the effort, plus small amounts of agricultural fertilizers also donated by industry. These donations being rewarded by tax deductions. In this 10-gram doses of spirulina to 30 million children would be about the same as for 1 day of seawater. Thank you.
I thank you, Professor Fox, for the very illuminating talk. The paper is open for the, I'm sure there are a lot of
Eventually, by a string of pilot plants where the quality problems are going to be solved one after the other.
Systems for village women for their income generation, and how we have to be manned by trained persons, and they will have to ensure the quality, etc., whatever parameters which are required, which cannot be met by the village women. And I think that's the best way to go about it. That requires a lot of coordination and planning.
Become this banal as growing wheat or something else or rice in this country, that every village will grow its own spirulina. But I'm worried.
Dr. N. Venkataraman, he was the formerly head of the Plant Cell Biotechnology Department in the Center for CFTRI, and Formulations. So, the topic is
distinguished delegates from India and abroad, ladies and gentlemen. At the very outset, spirulina in the next 5 years. As most of you are aware, spirulina, nearly 40 countries are producing at different levels. The huge Mexican plant was put out of commission due to economic reasons. Nearly 350 to 400 metric tons. India could have gone in a very big way several years ago, even though competent technology was available, but then it has been a late entrant, and in the last few years there has been tremendous development in production. Now, what I wish to say as in the next 5 years or so, in terms of the technological component, productivity modeling will be very important, because in spite of Mike Bharathidasan telling about the glories of nature, where the climate has got a very important component, and besides that, playing around with the nutrient media can have an impact on the chemicals. Now the photobioreactors, I just made a remark. I'm not saying the photobioreactor is not possible. That is possible, can be arrived at. And then the factors which have not been well understood, which affect both the quality and yield, are the photoinhibitions, the photorespiration, and photooxidation, and also the decay of spirulina cultures in very large quantities. Next one. Closed photobioreactors, I think I should not be talking, because we heard such a masterly and first-hand knowledge from Dr. Borowiczka, and I'm telling with reference to spirulina primarily, because I'm not saying photobioreactors are not workable systems. But even though a lot of question marks still arise, kinetic and physical-chemical parameters needs to be studied. The primary limitation, if you are going to use spirulina, can you move it a little more? System heats up, and it needs, of course, cooling. Spirulina strains have been isolated in India, so it is possible that we can use thermotolerant strains instead of cooling the whole system. And the other problem that commonly occurs in spirulina is the sticking of the cells on the tubes, which of course not only is a problem in cleaning, but also it curtails and safety. I think this is not a forum for me to discuss in great detail. Quite a bit in my paper, in a detailed paper I have given, which will be available to you. So here you find, when you are going to have an open system, now these are all the various challenges which are going to affect the ultimate quality of spirulina. And unless you have got a basic good material, you are not going to have your formulations. So you have, these are all the various parameters, the insects, the planktons, the microbes, the recycling schedules, the hygiene and sanitation you are having, and the kind of toxins inadvertently that we get. We may say generally spirulina does not have any toxic constituents, but being a material which can selectively uptake and biomagnify, you have to be very careful. Next one. I don't need for quality, safer quality. You find quite a bit of these export materials do not get accepted in the country, for which it is disdained, because there are so many qualities which are being stressed, including aflatoxins have been reported from spirulina material. And so you have to, I don't want to go through the whole lot of things, because there are a lot I want to say. And so there are stricter global safety requirements for any kind of food product which will be applicable to spirulina. Next one. And here I've shown some of the standards, microbiological related quality standards. Most of you are quite aware of it, so I don't have to say, excepting the great advantage of spirulina being grown in a very high alkaline medium, 9.5 to 10.5 or so, exclude a lot of pathogenic bacterial forms. In fact, we were the very first to establish that you can introduce contaminated spirulina. And here you have heavy metals. And this is again a very serious problem, because of biomagnification. Selectively they can uptake 10 to 20 folds more from the medium. And here you have the different kinds of standards which are but usually we talk only about lead, mercury, etc. But now arsenic, cadmium, etc. are also very important kind of metals which need to be monitored. Next one. Now, before I go into that, can I have the 2-3 slides? The biochemicals, and it can be grown in Cyanobacterial or so. But then the large-scale productivity systems are not very well established there. The advantage is spirulina systems have been very well tested for is saying, we have not reached any level of application. If you feel that Cyanobacterial is ideal, I accept it.
And the second thing is that, what is your target organism? A target pest which you want to
take.
take?
Any insect with BT can be applied like mosquitoes. Many of my friends from abroad will say that
we have to move on. Thank you.
That's all.
You have attended this culture.
Thank you, Mr. Looking forward. I'm a retired chemical engineer. Since about 6 years now, I've been cultivating spirulina on a small scale, experimenting along the basis of Dr. Gigi Bai's method towards artisanal spirulina production in the villages. But I would like to start by recalling a few basic chemistry as applied to spirulina culture. And then only after that I will show you the little experiments I've done. The first overhead, please. First I should say that of course I will refer to spirulina, but actually we talk about arthrospirulina. Remind you that the atmosphere contains CO2, and when it is in equilibrium, then CO2 will be lost to the atmosphere, and conversely, pH is above 10, CO2 will be gained from the atmosphere. Next.
Introduction, I would like to talk. There is so much on freshwater cyanobacteria, but I'm going to restrict myself only to marine cyanobacteria. And as Ripley Fox suggested, that the inputs here are much cheaper. We can use seawater, and therefore, for a country like ours, with a coastline of over 7,000 kilometers, I think this is really a better system to deal with than even freshwater cyanobacteria, because we have lots of water scarcity, and we are, especially the waste that are coming out of this, as Fox suggested, is going to cause problems. So the loss incurred by people who have invested their funds becomes all the more